Hello there, welcome back to GCSE Labs. This video will be on Required Practical 1 based on microscopes as part of the Cell Biology GCSE Science specification. Let's begin. Regarding stains, you must know this for your required practical. Stains are used to highlight or show the features of a cell more from color being added onto them. For plant cells, we always use iodine solution to color them, and for animal cells, we always use methylene blue. However, today's practical will be based on onions, and we know that onions are plants, therefore there will be plant cells that we'll be observing, so we have to use iodine solution as our stain. So let's begin. First of all, there must be a clean microscope slide ready. So if you remember from the first video for cell biology, we looked at microscopes and the different parts, and I did mention that there is usually a slide that comes with it, because this is where the specimen will be located and observed from. Secondly, in the middle of that slide, add a drop of water. So right in the middle, that's where the specimen will be. Thirdly, peel out some onion layers and use tweezers to obtain the epidermal tissue from one of those layers of the onion. So we're kind of taking out a layer from a layer when using tweezers for onions. Fourth, use the tweezers to locate the epidermal tissue on the slide where the water drop is. So exactly where the water drop is. Fifth, add a drop of iodine solution to this. So remember, we're working with an onion, that's a plant, we're observing plant cells, we use iodine solution. And these bottles usually come with pipettes, you know, this red component right here. And that's what's used to actually obtain out the iodine solution from the bottle. Now, you locate the cover slip, which is a thin piece of plastic or glass, and then you use this to prevent any air bubbles from being under the cover slip, as they would obstruct the view of the specimen. So you place the cover slip really carefully. It can be a square or a circle, but usually it's a square. Okay, so now this is what it should look like at the end of your prepared slide. So it should have the water drop, it should have the actual specimen, which is the onion, epidermal tissue. It should then have the iodine solution, and then this ends up getting mixed up with the water. And then finally add your cover slip, and this should all be placed on the slide. So just as a reminder from that microscope video, this is just what a microscope should look like. So there's the eyepiece lens up there, there's a coarse focusing wheel, the fine focusing wheel, objective lens, stage, and light source. Remember, we're looking at a light microscope. And you should just take this minute to familiarize yourself with how it looks and what its structure is, so we can move on to how we're going to use it for this practical. So using the light microscope, firstly, you position the slide on the stage and clip it on. Simple, just put the slide on the stage and you just clip it on. Second, you then select the lowest powered objective lens, which is the lowest level of magnification. Remember, the objective lens, they work and deal with magnification. Next, use the coarse adjustment knob. So this is the knob that moves the stage by a lot to elevate the stage to just below the objective lens. So we're just moving it up. While looking through the eyepiece, use the coarse adjustment knob again, but to move the stage downwards until it is in focus. So we don't want it to be too close to the objective lens. Then adjust the focus using the fine focus knob, which only moves the stage by a little bit, in order to get a clear image of the specimen on the slide. Finally, the magnification can be adjusted to get a higher level of magnification by increasing this in the objective lens, because remember we started with the lowest powered objective lens, but we can change this of course to a higher level of magnification using the objective lens by adjusting them. Now this is really important because you're going to have to know how to draw your observations. So when drawing your observations, you must use a pencil, make the drawing of your observations big, so they should take up at least half the space available. There should be no coloring or shading because we're trying to have a clear image of it all. Subcellular structures should be drawn in proportion with one another. For example, if the nucleus is bigger than the ribosome, then it should be drawn that way. Can't have the ribosome bigger than the nucleus. Include a title as well as the magnification levels that were included, so that way it's more accurate and more reliable. And finally, include labeling. So label where the different subcellular structures are, like the mitochondrion or the nucleus, the cell membrane, the cell wall. And here there must have been a cell wall because remember we were working with plant cells. So take a moment just to make note of how you can draw out your observations. Now, I hope you took what you need to from this video. The next video will be on specialized cells. Please do subscribe and have a lovely day. Goodbye.